Starting now. So these are actually the folder where we will start. This is the Fastqc file what I downloaded from NCBI. So what we will do, we will quality control this file, then we will trim this and we will mapping this. Now, if you open the terminal, so I want to check what is inside our folder. So this is actually our file. What we will do first, we will open the file and look into the file first. So this is a Fastqc file, and these are every read. This is the first read. These are our second read. These are our third read, and we have many reads here. And for every read, we have the quality information here. So now what we will do, we will process this file now. That means we will check the quality report of this file. For this, we will use the Fastqc tool. So how Fastqc tool works actually? Okay, before using the Fastqc tool, we will create our directory so that we can put all our output file in there. So for Fastqc file, we will create a directory QC. We will also create a directory for trimming and another directory for mapping. So if we continue with our Fastqc file now, so first the command Fastqc, then the file name, what we want to get the report, then the directory where the report will be saved. So we want to save it inside our output/qc directory. And now if we run this, it is actually running. Okay, the running is complete. So now if we want to open this file, we can go to our directory here. So this was our file actually. So now if we look at the report now, so first thing is our basic statistics. So this actually contains the file name, then the type of the file, then encoding. This means it actually a Illumina file, and the total sequences, that is the total number of reads, then total bases, and here actually the sequence length, that is for every read what is actually the length of that, and here is the GC percentage, so it is 48, that is it is quite good. For eukaryotes, we actually expect the GC percentage within 40 to 50. So the cross sign, that is, we can see here some cross sign. This cross sign means these uh, these matrix actually fails. So what we need to do, we filter the this file, or we need to trim this file to fix this failure actually. So here, this is actually important, the par base sequence quality. If we look at here, these are the box plot. So what we know about the box plot, that is, if we consider this box here, so this is actually the minimum read at that position. That is, these are actually the positions. So we had 190 position for each read. So for 190 position, we have one box plot for each position. So if we take this, that means it is for 185 position, and this represent the minimum quality score. Then this is our first quartile. Then this red line consider the median. This is our third quartile, and this is the maximum quality score. And here the blue line, this actually represent the mean quality scores. So if we here are few region, that is, this region is colored with green, this with orange, and this with red color. So in this region, that is, the quality score greater than 28. So this region, these are actually very good. And what is inside the orange region, that is 20 to 28, this region is reasonable actually. And if the quality score lies less than 20, it will uh, shows the failure error. That is, we need to fix it actually. So here are another thing. There is the par base sequence content. That is, we know we have four bases, that is A, C, G, T. So what we see here, we have position in the x-axis, and these actually represent different color. There is a percent of T, percent of C, percent of A, percent of G in each position. So what we expect uh, all these will be parallel to each other. So they won't have much difference. So for this read, actually, first 15 reads, they have a fluctuation of them, and at the last they have a little fluctuation. So to fix this, we can drop few bases from first and drop few bases from last. The GC content actually it actually what the x-axis contains the mean GC content, and the y-axis, this is the number of read actually. So this actually the theoretical line that we should have, and this actually our GC percentage line. Here these sequences are over represented in the reads. These sequences need to be deleted, need to be removed from the reads. Twisted the next step, and adapter sequence should be removed. So now if we start our trimming, so what we need to do, we can drop some bases from first and drop some bases from last. So we can drop first 15 bases and we can drop last 3 bases, that is here. And for trimming, we actually expect the quality, that is after trimming every box, that is every quartile box uh, we will be within at least the orange region. So this should be removed actually. What is lies in the red region. So for this, if we consider the score 28, I think it will be okay for this. Okay, let's see what happens actually. Hmm. Here are the thing, if you read this, you will understand, no problem. And I'm just copying the command from here. So first thing is this is our tool trigonometric, and SE represent this file is actually single int, and threads, this is the core, how many CPUs the code will use actually, and these are file name, what we will trim, and these are our output file name, where we actually save the file, that is inside our output trimming directory, and these are the file name, that is trim.fastq.gz. Then illuminate clip, so we want to clip the adapter sequences. So for this reason we have a file actually, so that contains few adapter sequences. This was collected from the Illumina site, and what we expected, that is the leading score. Leading score should be, we set the leading quality score as 28 and trailing quality score as 28. Then we want to drop the first 15 bases and we want to keep the last 172 bases. So, and the minimum length of each read, what we will consider as 36 actually. So, okay, no, the command broken actually. So, if we run it again, Uh, the problem is this command were separated, so it showed error. Okay, now it's okay. So if we run it, Chemometic is running now. So look at here, what we will have the output. That is, this actually our output file. So what we will do, we will again quality control that file. So first QC, then our output file name, then the location where we want to save the output. So if we run it now, okay, so it's finished. If we go to our directory, there is this is the file what we have got now. Now, if we look at the files, these are almost okay. So we almost fixed everything for this file and we can use it for our next step. So if we look at here, so almost every quality score are inside our green region and the minimum bases are inside the orange region. So it's very good. And our par base sequence content. So now we can check they are almost parallel. That is, they have small fluctuation among them. And our GC content, they mess with our theoretical line actually. So the blue line is our theoretical line, and this one that is red line is actually for the GC count. And we almost fixed our over represented sequences here. So this is the percentage. So how much time the sequences was appeared, and we removed our adapter sequences. So if we look at the previous file, you can compare with that file, this one. So we had actually the adapter sequence. What adapter sequence we had? Uh, this one, that is the yellow one, that is the next error transpose sequence, and this one that is actually poly A sequence. These are the type of the adapter sequences. So we had completed our trimming. Now we can process. Now we can proceed for the mapping section. So for mapping, we will use the tool HISAT. For HISAT, we need to first we need to build our reference genome. So we we will simply use the same first the same file, that is this was sorry, this was our Fastqc file, and we will use the same sequence as a faster file here. This is for simplification, but uh, when we will map, we should use some a genome. That is, we can use the AG19 or AG38 genome. That will be for whole genome sequencing, or we can take exon for whole exome sequencing, something like that. So we are using this one for the simplification actually. Just need to for understanding the process. So what we need to do now, we will build a reference for this reference genome. So for building the reference, so here is the command. This is the file. This is our reference file. And we need to mention the directory where we want to save it. Mapping. Then suppose we want to mapping it with genome index. So this reference genome will be indexed as genome index. So if we run it now, it may take few minutes. So after genome indexing, so we can start our mapping. That is, we set 2, then dash p. This means the threads, that is number of CPUs what will be used actually, and dash x. After this, we should mention the genome index location where we saved our index actually. Then dash capital U. After this, we should mention our file name. So this was the file name what we processed. That is that we trimmed this and fix our errors. And this is our output file name. It will be in the same format, and we will save a summary file in the same directory actually. So if we copy it, okay, this is still running. Okay, so it's finished now. So if we want to check what it did actually, inside the mapping directory, it generated these 8 files. So it starts with genome index, what we used actually, genome index .1.ht2, and it always build 8 indexes for the reference genome. Now if we paste our command here, so we explain this command, what this means. If we run it now, it's running actually. So here we get this output actually. So this is the number of reads and how many times it aligned actually. So 33.02% aligned one time, and 66.98% reads aligned greater than one times. If we okay, so if we want to check what is inside our SAM file, we can use the SAM tools to view our files. That is, so we want to see it as the SAM file, and it was inside our mapping directory. Then SAM tools, view, and these are these are output file that we want to view actually. So if we okay, so it's actually my fault. So we just want to view what is inside the some files. These are the row actually. The first time we use the B prefix, B command, so that is for that reason we got the output in binary format. That was actually unexpected. So these are the text format. We can view it like this. So in this way we can complete our mapping. After this we can proceed for the variant calling, and we will explore the variant calling later actually.